All right, good afternoon, everybody. Thanks for joining us today. This is our last call for the year of 2020. Um, we always have a number of people hop on in these first few minutes, but we'll go ahead and get started right at 1:30. I'm particularly excited about today's presentation because this is a story I've been kind of following a little bit for the past few years, and they recently published some more work on it. So something really interesting and something we probably should all be keeping an eye on is this parasite that was introduced via the Burmese python, but is now having an impact beyond the range of the python. So I'm really excited to hear more about that from Dr. Melissa Miller, who is a research coordinator with the UF-IFAS crop ducks program down in South Florida. And then we have a couple good updates for you guys today once we wrap with that. So with that, I'm going to turn it over to Dr. Miller to jump right in.
Okay, thank you, Emily. I appreciate that. Let me go ahead and share my screen here, see if I can do that correctly.
Perfect.
Are you guys seeing presenter view or are you seeing?
You're good to go.
Oh, okay. Interesting. Okay, let's see here. Let me just minimize the little screen here. Okay, so thank you for the introduction and thank you all for joining me today. So I'm always happy to discuss pythons and parasites. And today I'm going to be discussing how host-parasite dynamics are altered during biological invasions. So specifically, the talk is going to focus on a lung parasite that was introduced by Burmese pythons. And we found that this parasite has spilled over from pythons to, unfortunately, now infect 13 species of Florida's native snakes. So I'll be showing data to support that native snakes are highly susceptible to infection, and it's facilitated the spread of this parasite to native snakes that are outside of the python's range. So the parasite now does not need pythons to spread. It's actually doing quite well without them. And lastly, we'll examine how this parasite is so successful at infecting such diverse hosts and how the parasite is capable of altering its oral morphology in order to do this. Okay, so as we all know, biological invasions are increasing globally, and much of the world experiences a mid to high level threat from invasive species. And if we look at the map, mid to high level threat areas are shown from yellow to orange to red, respectively. And 42% of our threatened and endangered species face risk due to, in some way, shape, or form, due to invasive species. So one of the ways that invasive species can threaten native wildlife is through introducing parasites and pathogens. And when these parasites are introduced, a study showed that 78% go on to infect hosts within the parasite's introduced range. So when this occurs, the parasite is often more virulent. So parasite spillover is basically, it's just the spread of an introduced parasite to native taxa within the parasite's introduced range. And a good example of this can be seen with a nematode parasite. So this nematode was imported with the Japanese eel to the UK, and where its parasite now went on to infect the native European eel. And when it did that, the nematode actually exhibited increased intensity and survival, and it grew, it had faster development, and you can see a mugshot of it here on the right, within the native European eel. So it actually was more, the native European eel was a more competent host for this newly invading nematode. And unfortunately to the native European eel, it suffered enlarged swim bladders and poor body condition from the infection from this introduced nematode. So parasite spillover can lead to some pretty detrimental effects in taxa within the parasite's invaded range. So something that's very interesting to me is how do these introduced parasites, they infect such a diverse array of hosts, and how are they so successful at doing that? And we know that parasites may exhibit host-specific phenotypic plasticity. So for example, if we look at this mite here, a study was examining this, and morphologically, the mite appeared to be 5 different species. When looking at the genetics, it was 1 species. So this particular species of mite was able to show a lot of phenotypic adaptation that was specific to the microenvironment of its host. And this can help increase the ability of introduced parasites to infect novel hosts. So parasite introductions provide an opportunity to investigate parasite infection dynamics and explore how a non-native species, in this case the parasite, adapts to a novel environment. So the questions of interest for us were, for one, is parasite spillover occurring? And how competent are novel hosts? So how well does the parasite do if it infects novel hosts? And then how do introduced parasites adapt to infect hosts within their invasive range? And to address these questions, we examined the invasive Burmese python population in Florida. Burmese pythons unfortunately need no introduction for this group. They were introduced to South Florida through the pet trade, ultimately, or through either unintentional escapes and intentional releases, probably augmented by hurricanes. Pythons became established and breeding in the wild around the mid-1990s, early 2000s. So now pythons can be found south of Lake Okeechobee from the west coast to the east coast and down to the upper Florida Keys. And as of October, FWC reports over 12,000 pythons have been removed from Florida. So we know a lot about the direct impacts like through predation, for example, the mammal declines that pythons have caused in Everglades National Park. And indirect impacts are often less studied. They're not as obvious, I guess I could say. And ectoparasites have been surveyed, but endoparasites really have largely been left untouched. So we don't know a lot about the endoparasites and risk pythons pose through potentially introducing them. So I'd like to take a second just to tell a little backstory about how the study came about and give credit to the man that set it in motion, which many of you may know, Skip Snow, who's now retired. He's a wildlife biologist for Everglades National Park, and he started all the research on Burmese pythons. And in 2011, I was getting ready to start my dissertation at Auburn University, and I was down here. I'm actually living in the Keys, in Key Largo, and I was a volunteer for the park's Python program, and that's where I met Skip. And Skip had been finding these penistomes, and this is one of the very early penistomes that in this picture here you can see Skip was necropsying a snake. And we knew, there were penistomes, which are endoparasitic crustaceans that they infect the lungs of reptiles. About 90% of their hosts are reptiles. And they attach to the lungs and feed off host blood. But the question was basically, well, is it native or non-native? So we knew the penistome belonged to the genus Reliatiella. However, identifying penistomes beyond genus requires molecular analyses. So it can get really tricky due to some overlapping morphological traits. And the question we needed to know was if this lung parasite was or had been introduced by Burmese pythons, in which case we expected to be Reliatiella orientalis, because that's a penistome known to infect pythons in their native range in Southeast Asia. The other option is it, it could be Reliatiella bicaudata, which is reported to be in North America and is known as the southeastern snake penistome. So we knew we had a Reliatiella, we didn't know what species it was, and therefore we didn't know if it was native or non-native. So we needed to run molecular analyses to identify which species we were dealing with, and there was several potential outcomes for the molecular analyses, which included the following. So one, we could have both species present. We could have the non-native R. orientalis and the native southeastern snake penistome R. bicaudata, in which case we would expect to recover 2 clades from our morphological or molecular analyses that represent these 2 species. Alternatively, we could have the non-native Reliatiella orientalis only, where we'd expect to have all of the Reliatiella collected from Florida would cluster with Realitia orientalis reference samples that we were able to get from their old world origin, from their native distribution, and we were able to get these off GenBank. And lastly, we could have just the Realitia bicaudata. And in that case, we'd expect to recover a single branch for North American Realitia remote from Old World Realitia samples from GenBank. And I wish, it's probably as much as you all right now, that there was a common name for Realitia orientalis, but there is not. So bear with me on that. You're going to hear it a lot today. So to answer these questions, I needed a lot of snakes and a lot of parasites. So I conducted a lot of road surveys. I worked with folks from the Python removal programs to get a lot of pythons. And there was an army of volunteers all throughout the Southeast that would provide me with dead snakes. So native snakes, as I just mentioned, they were collected as fresh roadkill because it was determined that wasn't going to affect the outcome of the study. And obviously, being a herpetologist, and I like snakes, and I did not want to go out and kill snakes. So it was great to know that I could use fresh roadkill and make use of those salvage specimens. So all in all, we got 523 of 26 different species of native snake. And Burmese pythons, through the python removal programs, doing road surveys and volunteers, we were able to look at 1,003. So snakes were dissected and pennistomes were collected, and here's my amassing pennistome collection during my dissertation. And the snakes were, after they were dissected, were all put into the museum at Auburn University Museum of Natural History, and many of them on loan from the National Park Service. They're collected in Everglades National Park. So for molecular analyses, I'll kind of skip over this quickly, but we did a helix DNA extraction on some pennistome tissue. We did a PCR amplification of 2 genes, CO1 and 18S, and the DNA was sequenced, and chromatograms from both forward and reverse reads were assembled, and sequences were aligned. So I'd mentioned there were some old world samples available from R. orientalis from their old world distribution, and those were available on GenBank. So those were included in our molecular analyses, and those were 18S. We had one sample that was actually labeled as R. orientalis. We had access to 2 samples of Realitia that were from the right place, but we didn't know if they were orientalis or not. Turns out later, spoiler alert, they were, which worked out in our favor. And then we had, for CO1 samples, there was R. orientalis from Australian snakes. And this is interesting because R. orientalis is also considered to be introduced to Australia, although they don't know the pathway. So there was some recent work by a really good ecologist, Crystal Kelly here, and she had many samples available on GenBank of CO1 for this species that we could use in our analyses. And then we included another Realitia, Realitia hebetahemata. Yes, they all have great names. And we only had one of these, but we wanted to include this to test our ability to differentiate different Realitia species when we recover our lineages. So Realitia bicaudata was not available on GenBank. There were no samples that we could access through GenBank, but it was reported to infect snakes in southeastern U.S., and we collected a lot of snakes in southeastern U.S., so we fully expected we'll just find that one ourselves. And any monophyletic lineage that excluded published R. orientalis sequences could be attributable to bicaudata. So for our phylogenetic analyses, we perform them on a concatenated data set, which just makes use of data from 18S and CO1 genes. And for the outgroup, we used another pennistome species. We also included, as we were going through all these snakes, we were finding the two species of native pennistomes that are native to North America. So we found Curocephalus corictatus and Porocephalus crotali, which gets into pit vipers. And we included these in our analyses. And we did a best fit model of evolution using AIC in a Bayesian phylogeny inferred by Mr. Bayes. And eventually we conducted, at the end of the day, a 50% majority rule consensus tree created using the consumption of Mr. Bayes. So here's our phylogeny for the pennistomes, and let me see if I can get the laser pointer here. So we recovered 3 distinct genetic lineages of 3 genera. So we have the Reliatiella shown here in purple, and then we have Porocephalus, which was again the native pit viper, native to North America, native to our pit vipers. And then we had Chirocephalus, which is native to our colubrid snakes in North America, shown here in the orange. And this is our Reliatiella hemata, which I'll discuss here in a second. Oops, let's see here. Oh no. Just had a little frozen screen issue that I'm going to try to work through here. Bear with me. Oh, okay. Interesting. Yeah, I apologize. I'm having of course, during a presentation, but a Microsoft PowerPoint not responding message here. So let me see if this will work with me or not. Okay, I think we're back on. Can you all see? I apologize for that. Can you all see the screen still with the phylogeny?
Yes, I think you're good.
Okay. All right. Never know when that stuff's going to happen, but you just have to roll with it. So, okay, so we recovered a single clade for Reliatiella, and that was the purple. So that includes pennistomes collected from pythons and from Florida native snakes. Also within that lineage are the Australian Reliatiella orientalis samples, and most importantly are Old World samples of our orientalis from the native range. So there was no structure within the lineage. This indicates we're dealing with one species, and based on the samples that came out, including the Old World, the Australian ones, and all the Florida native snakes and pythons, we attributed that to Reliatiella orientalis. And the Reliatiella hemata, so that's this little pink lineage at the bottom. It failed to cluster with the Florida and Old World and Australian samples in the purple lineage, and that shows that we were able to detect, our analyses were able to detect a second species of Reliatiella had we have found it. So we did not recover Reliatiella bicaudata. So kind of in summary of the molecular analyses, we found evidence to support a single species, Reliatiella orientalis, infecting native snakes and pythons. And the Reliatiella from Florida native snakes and pythons did create a single lineage with reference to the Old World R. orientalis specimen. Reliatiella bicaudata, we do not consider a valid species. So we had 523 native snakes, southeastern snakes that we looked at, and although absence isn't proof of non-existence, no lineage attributed to this species was recovered. And there are, looking back at the original description, this species was described from some specimens, some snake specimens in a zoological setting, so they weren't necessarily collected from the southeast. They spent time in captivity. So we think that, along with the lack of morphological traits to differentiate Reliatiella or pennistomes in general, definitively beyond genus, that these Reliatiella bicaudata was likely misidentified. So we know Parasite, Reliatiella orientalis, is from Southeast Asia, and we know that's what we're dealing with. And we found the parasite has spilled over to infect native snakes in Florida, but now the question becomes, how competent are native snakes as hosts? So how bad are they getting this parasite? Or does the parasite do well in native snakes at all? So for host competency, basically, it's just how well does the parasite do within the host? Can it complete its life cycle and reproduce? And some metrics we used to examine host competency included prevalence, which is the percent of a sample infected, and infection intensity, which is the mean number of individual parasites per host, the size of adult females. So this one is interesting because female size for parasites particularly is a proxy for fecundity. So the increased size the increased egg output that's going to be possible. And then we looked at the proportion of adult females to determine the percentage of the pennistomes within a host. How much of that percentage consists of reproductive females, which are responsible, obviously, for getting the eggs out into the environment. So pythons were captured from throughout the range through the help of lots of road surveys and partners. And DOR snakes, as I mentioned, those were collected and salvaged as fresh roadkill. And they were collected from areas sympatric with pythons. So the counties in red, we designated as the core of the python population, so that would have the highest python abundance. The counties along the invasion front in yellow are areas where pythons are present, but they're less abundant compared with the core. And our invasion front is kind of a conservative estimate, honestly, with the counties at the northern range, but we wanted to be err on being more conservative than not when designating that, because obviously invasion fronts can never be known definitively. And we collected snakes from regions allopatric with pythons because we were at this point starting to see some infected snakes in the core and a little bit in the invasion front, and we wanted to know how far north this parasite had traveled. As well, we were looking for the southeastern snake pennistome for our molecular analysis. So we collected 523 native snakes, so 3 families, 17 genera, and 26 species. We wanted a really diverse representation of the snakes in Florida. And again, we had the 1,003 pythons. And we collected the parasites, and from native snakes, we collected 1,083 individual Railitaila orientalis. And from pythons, we collected 255 of this parasite. So if we look at the map here, the red again is the core of the python population. The orange is the invasion front. The gray is regions of allopatry with pythons. So there's no known established population of pythons in the gray area. And the yellow squares represent a native snake infected with R. orientalis, and the black squares represent a native snake that was not infected. And having to show the map at this scale to kind of show the range for our sampling, it does cause a lot of overlap with some of the squares, so I apologize for that. But we did find 13 native species infected with this parasite that was introduced by pythons. We found 3 snakes that were infected in a region of allopatry with pythons, so in Lake and Polk counties, and the northernmost infected, or 348 kilometers north of the northernmost infected python. So that was pretty exciting because that let us know that this parasite, that native snakes are so susceptible to infection that this parasite can now spread independently of Burmese pythons. And we sampled from Alachua northward, and Alachua County, and we did not find any infected native snakes. Although a recent paper that came out in June and found that a banded water snake in Alachua County was infected. So there's just a few years later, they were able to find that parasite has now been documented in Alachua County. So let's go ahead and take a look at the results. This is a prevalence. So for prevalence analyses, you're going to see some weird acronyms at the bottom. So let me talk you through those real quick. So we have native snakes were grouped by foraging ecology to use as a proxy for diet. So to identify which snake species are most susceptible to infection is one of the reasons we did this. And another is to identify potential intermediate hosts involved in the life cycle of R. orientalis. So this parasite has an indirect life cycle that involves multiple hosts, and it is unknown in the literature in its native range, and it's unknown in its Florida range. So we wanted to look at what these snakes are eating and what can that tell us, if anything, about the intermediate host that may be involved in the life cycle. So the groupings were based on foraging ecology, included I still have that on there. The terrestrial ambush predators. So this was our cottonmouths and pygmy rattlesnakes. And then our terrestrial wide foraging non-constrictors, which are racers, coachwhips, and indigo snakes. And then we had our aquatic non-constricting frog eaters, which were our basically water snakes, banded water snake, mangrove water snake, and garter snake. And then we had our terrestrial wide foraging constrictors, which were our corn snakes, rat snakes, and king snakes. And last but not least, our aquatic non-constricting fish eaters, which was the green water snake and the brown water snake, and of course python. So if we look at the black bars, they represent groups that did not differ significantly from one another. So if we go back to this, and not asking anyone to remember these acronyms here, but if we look here, we see that our cottonmouths, pygmy rattlesnakes, racers, coachwhips, indigo snakes, and our frog-eating water snakes did have the highest prevalence compared relatively to our rat snakes and corn snakes and king snakes, which were in this grouping. And our fish-eating snakes did not differ significantly from pythons in terms of prevalence, and that's kind of indicative of the fact that fish likely are not involved in the life cycle of this parasite. So I'll have a little bit more on that later on of how this relates to the life cycle. So looking at the figure for infection intensity, we have the number, which is the number of parasites per host. We have a mean infection intensity of R. orientalis on the y-axis, and native snakes are grouped by region, either the core or invasion front, depending on where they were captured on the x-axis. For pythons, the core and invasion front are grouped together because of a small sample size of R. orientalis, which was n equals 2, recovered from pythons in the invasion front. So infection intensity of R. orientalis differed significantly among groups, with intensity in pythons significantly less than intensity in native snakes collected in the core, but not distinguishable from the native snakes collected in the invasion front. And the black lines throughout the figures all represent groups that do not differ significantly. So just to give you an idea of the range of pennistomes that we would find as far as infection intensity, they range from 1 to 77 pennistomes per host. And if you look in these vials here, those were all collected from one individual snake and a coachwhip that was heavily infected with all of those in its lungs. So that was very concerning. While pythons, on the other hand, pennistome infection ranged from 1 to 15 pennistomes per host. So looking at body length of adult female R. orientalis, and again, the body length of females was examined as a proxy for egg production output fecundity. So female pennistomes reach significantly longer body lengths in native snakes collected from the core region than in pythons, but they did not differ compared to native snakes from the invasion front. And here we're looking at the proportion of adult female R. orientalis. So what proportion of the pennistome population in a host is made up of basically reproductive adults. And here we see some pennistome eggs in the picture. And if you look, you can see some embryonated eggs in there. And even at that stage, they still have the little hooks. And it's pretty interesting. So what we found here was that native snakes from the invasion front varied significantly from pythons, native snakes from the invasion front and the core both differed and had larger proportion of adult female R. orientalis in them compared with pythons. So native snakes are more competent hosts, is what our data shows of this parasite than pythons, and they exhibited a higher prevalence and intensity from snakes in the core versus pythons, and the pennistomes were larger body size for snakes in the core, and then adult female pennistomes occurred at a higher proportion for both snakes in the core and invasion front compared with pythons. And native snakes are more susceptible to R. orientalis infection likely because this is a novel parasite for snakes in North America. So there are other genera of penistomes, but this particular penistome is a new genus, and it's not been encountered by our native snakes. So there's a lack of shared evolutionary history there, and they're not co-evolved. So in cases of that, the host can be immunologically naive and suffer more adverse impacts due to infection. And we found approximately half of the snakes in extreme southern Florida, Miami-Dade and Monroe counties were infected, so 13 of 26. And actually, a friend of mine out on the West Coast, Matt Metcalf, he found that eastern diamondbacks, he found infected out there too, so that brings the total up to 14, actually, if we include his work. So this parasite has spread beyond the range of pythons, and this is very concerning because native snakes were infected north of the invasion front, and if you think of native snakes being able to now pass this parasite amongst themselves, thinking of like the garter snake, which has different, species, subspecies. This is the range of it shown throughout North America and Canada, and actually goes into Mexico too. But this is a lot of these snakes are very common species that are widespread and ubiquitous, and it's concerning to think that this, might not be just a Florida problem, that this could affect native snakes well beyond Florida. So I mentioned the parasite has an indirect life cycle, and it can have an impact to intermediate hosts as well. So this life cycle is unknown in its native and introduced range, but our 2018 work showed a shrew, a mammal, could be in an intermediate host, and that was the first time that was discovered. So we know mammals can be involved. Our current study supports strongly that frogs are likely intermediate hosts and play a role in the life cycle. And our current research, we're looking at some invasive lizards that also can get R. orientalis. So, and what are the implications of that is kind of what we're looking at next. So both primary and intermediate hosts are at risk. So all these diverse snakes are being infected, and the parasite is highly competent. And now we know, as I mentioned with the current work, that invasive lizards are also capable of being infected, and what are the implications there? So how is this pentastome so successful at infecting such diverse taxa? So we explored morphological plasticity of the parasite to see if that's what's influencing its success. And we used a shape analysis, a geometric morphometrics of R. orientalis, looking at the oral morphology of R. orientalis collected from different host species to see if that may have something to do with it. So we looked at Burmese pythons and compared those with cottonmouths, salt marsh snakes, and garter snakes and racers. And we looked at 44 penistomes collected from 15 host individuals. So female penistomes were used in the analysis, so we were only using one sex. Penistomes were cleared so that we could see the stereotyped portions of these are the hooks right here. They have two sets of hooks, and then oral cadre, or you think of it as a mouth area. And this is just a little shout out to an undergrad that I worked very closely with here, and she did a lot of necropsy work with me. So to do these analyses, we placed our landmarks on the insertion of the hook, the curve of the hook, the esophageal valve, and the interiormost point of the oral cadre. So basically just the oral structures. So we performed a Procrustes ANOVA and a CVA, and to test and visualize variation in head shape, and we generated a CVA and a transformation grid to visualize this morphological variation. So if we're looking here at the CVA, we see that geometric morphometric analyses yielded 4 distinct morphotypes in regard to R. orientalis, the oral morphology. So we had a distinct morphotype, or I guess oral morphology, that was required to infect pythons, another distinct one for cottonmouths, and another one for terrestrial cleaver constrictor racers. And then a similar oral morphology to infect garter snakes and mangrove snakes. So habitat, kind of aquatic or terrestrial, was one of the probably the most informative things separating the morphospace amongst these pennistomes. And the transformation grids just show the direction of change and how that varied by host. So geometric morphometrics analysis revealed distinct parasite morphology according to host taxa, and there was no genetic variation among pennistome sequences. So it was kind of a case similar to what was found in the mite example I presented earlier on in the talk. And this parasite is capable of altering its morphology, its oral morphology, to adapt to different host species. So it's not just exploiting a naive host, it's capable of actually adapting its morphology to increase its success based on which host taxa it's infecting. So this could hinder morphological species identification, which is the last thing pennistomes need in general. So in summary, pythons have introduced a non-native parasite, and this parasite does very well in native snakes. It's capable of adapting its oral morphology to increase its success in infecting native snakes. And importantly, the parasite can spread to locations without pythons. So now it can spread independently of pythons, and has, it demonstrates the invasive species can have impacts that are very far-reaching. And as I said before, it's not just a Florida problem if this thing can spread. Pythons are limited by climatic and physiological factors as far as how far north they can go, but parasites are not. So it is a big concern and something that needs to be watched for native snake populations. So I'd like to thank a bunch of folks. As always, it takes a whole bunch of folks to get work like this done, and especially with invasive species. And with that, feel free to reach out to me if you have any questions or like to discuss anything further, and I'll take any questions if I have time. Thank you.
Awesome, thank you so much. So we have some time to take questions. Feel free to type any questions you may have in the chat box. Dexter's kicking us off. Could the failure to detect two R species, I'm not even going to try that one, with only one species suggested as occurring in Florida be the result of lineage sorting if the two R species are closely related? I'm not sure two conserved mitochondrial markers could differentiate the species.
Yeah, so we could have looked at more genes, definitely. I think we felt pretty confident in that because that was one line of evidence that we had, was that we failed to detect the Raillichella bicaudata, but we did detect the other Raillichella species, which you would assume would also be closely related to Raillichella orientalis. And if we would have found the Raillichella bicaudata, so we looked all the species, the 13 species that were infected in South Florida and a little bit beyond the python invasion range. Those same species, those same 13 species that we sampled in northern Florida, in Alabama, and even bits of Georgia, never had Raillichella pennistome in them at all. So that makes us pretty comfortable just looking at the spatial range of it, that we never found a Raillichella outside of an area where there's pythons, or just outside of where there's an area where there's no pythons. That we were dealing with something that was brought over by pythons. And this parasite is known to be use snakes as a primary host, including the Burmese python.
Great. So I'm going to expand on the next question a little bit, which is it a fatal infection on small-sized or young snakes? To add, kind of generally, what do you have a sense of what this means for our native snakes as far as what the impacts are on their survivability or health going forward?
Yeah. So actually, I have I should have put this slide in there, but if I'm still sharing. So this is kind of a general list of pennistome impacts to the host, and most of this we know from literature on the effects of penistomes on co-evolved hosts, and particularly in lizards, like the Mediterranean gecko, for example. There's been some work there. And so obviously a parasite doesn't really, its main goal is not to kill the host because it wants to exploit it and get resources, right? So, but there are negative consequences of that. And in co-evolved hosts, these are some of the things: infection, they can cause asphyxiation through blocking the trachea. You can see here one thing pulled out of a garter snake that, that I found. And so when that happens, you know, the effects are bad enough, but when you get a species that's not co-evolved with it, the literature shows that it's often worse. The effects are often worse in these cases because they're immunologically naive. Now, over time, what will happen? You know, that time will tell. I mean, it's hard to gauge what will happen. But we do know that if you have increased metabolic demands, for example, you're probably not going to be, for the species of snakes that are heavily infected, they're probably, if your lungs are clogged up with penistomes, for example, just that impact alone, you're probably not going to be able to compete as well with other species, and you're probably not going to be able to escape predation, as efficiently as you would without this infection. So it needs to be examined, directly to find out what exactly are the impacts to snakes in Florida. But it's easy to see that, they would be expected to be negative.
Sure. Is there any form of treatment?
So in a veterinary setting, they can endoscopically go in and remove a penistome from the lungs of a pet snake. Obviously, not feasible for the wild. There is a closely related sea lice are closely related to penistomes, and they aquaculture will do some type of UV radiation to kill the eggs, and it's never been looked at with penistomes. But it's there's not any clear cut out to this, to be honest, and it just kind of shows with invasive species there isn't always an easy way to rectify the problems that come with species being introduced.
Sure. Is there anything, is there any way that this network of statewide CISMAs can contribute to y'all's work? Is there anything you need at this stage as far as looking at snakes beyond the range of pythons, or kind of what are y'all's next steps? And are there any ways that, a network of informed folks can contribute to that?
So I think, just monitoring the spread and just understanding more about the direct impacts to native snakes. So if you see a really emaciated snake or a sick-looking snake, and something interesting about penistomes is that they can try to exit the body of the host when the host is deceased. So if the host has been deceased for long enough, the penistome can detect that and actually try to come out through the trachea or the just through the body with those hooks. It can tunnel out. So it's pretty amazing thing. So like if you see a dead snake, you could, especially one that's been unfortunately run over and opened up a bit, you may be able to see these penistomes. They're easily visible with the naked eye, the adults anyway. And just keeping an eye out and documenting that would be helpful. And of course, collecting any specimens, putting them in your freezer, but that varies in the person. Takes a special kind of person with a special kind of spouse to allow them to do that. But that's always helpful, and just reporting that, and mainly, just reporting non-native species in general using the 888 I've got one hotline or I've got one.org. Just so we can prevent invasive species more from becoming established and bringing over additional parasites.
Sure, great, thank you. So I have one last kind of question for you, and then we'll move on. I'm not seeing anything else come in. But again, you can reach out to Dr. Miller if you have any additional questions. Since we have you, I just wondered if you could share a little bit for folks who may not know about kind of what the Crop Docs program is. I follow you guys on social media. It's I'm really interested just because I love herps in general, so I think y'all's work is really cool. And I realize not everyone may be kind of familiar with the program itself.
Yeah, so, well, the crop talks are kind of twofold. We do a lot of work with crocodilians, with crocodiles, as far as crocodile nesting and monitoring, also with alligators. And then we have the invasive side, which is the side that I do the research coordination for, the invasive species projects. And for that, we do a lot of work with research, monitoring, and analyses, kind of population modeling with tegus, with Nile monitors, with Burmese pythons, with caiman, and we work a lot with our state and federal partners on these projects, and we do our research in an adaptive management framework so we can help provide sound science and the best available science for management decisions.
Awesome. Well, thank you so much for joining us and providing this really interesting presentation on that work y'all are doing. It's so important. And for folks who, again, aren't really aware of Crop Docs, I encourage you to kind of look at their website, or they're on social media as well. They have some really cool projects going on. And it's really great research being contributed to the whole of invasion science in Florida. So I am going to pop my screen back up, and we just have a few updates for you guys today. So we should be good. Let me get my little boxes up here. Okay. So I wanted to this is usually my closing update, but I wanted to start with it today. As I mentioned, this is our last call of 2020. We take a break for November and December, and we're starting to put together 2021's call schedule. So we'll be back in January with some really neat work coming out of the University of Minnesota, where they are looking at common names of invasive species and how what we call those species may impact on kind of a social scale the work we're doing. So they're looking at the impacts of common names of invasive species, and it's really neat. I had a chance to talk with Angela last month, and so this is going to be a really cool presentation about how they are looking at kind of just how we talk about this issue with the public and with different groups. In March, we're going to have Sarah Funk with FWC provide an overview of the FWC Non-Native Animals Program. So as you guys know, they have been working on updating some of their invasive species rules. So we'll get an update on that, as well as just kind of who they are and what they're doing. I think it's a good time just to hear from them and understand better what the FWC Non-Native Animal Program is all about. So with that, the questions I have for you guys today are, what would you like to hear about in 2021? So if there's any topics that are emerging for you, or just that you feel like it's time to get a refresher on, please let me know. You can go ahead, if you have ideas now, you can type them in the chat box, or you can always email me suggestions. If you have a topic but you don't have a contact for it, that's fine. If you do have a contact, that's helpful. And also, do you have work you would like to present? So if you're working on something that you think this group, that would interest this group or be beneficial for this group to hear about, please let me know. So we will be working on filling out the schedule over the next couple months. I wanted to briefly kind of provide an update on kind of National Invasive Species Awareness Week / Weed Wrangle stuff. So go ahead and save the date, sticking with our last week of February every year. For 2021, that is March 22nd through Sunday, February 28th, with kind of the Saturday Weed Wrangle Day being Saturday, February 27th. So this will be Florida Weed Wrangle Week 2021. Now, it's complicated by a few things. So I'll actually switch those bullet points. First, I'll give a little update on National Invasive Species Awareness Week. I was on a planning call for that earlier this month, or late September. My dates are all kind of jumbled right now, but and really it's kind of up in the air what they're going to do given the ongoing issues with COVID-19 and some other things. So they're kind of still working that out. NASMA also just had their annual conference, so I imagine we'll hear more about NISA kind of as they get in the clear of that big effort. I know they have another conference coming up as well. So I'll let folks know what's happening with that, but so far I really don't have much word other than they're kind of trying to figure out how they want to go forward with those efforts. So for our part, I am really planning on doing a big push for stay at home, kind of manage the invasives in your own landscape efforts. I'm looking into providing some kind of prizes / awards for each of the 16 CISMAs for having connecting with folks via social media and saying, hey, work in your own landscape, let us know, and then and win, you know, the possibility of winning different prizes. So I'll let you guys know more about that in November. But I really, after kind of thinking about it and chatting with some different people, we just still don't know what winter is going to look like. We don't know what spring is going to look like with the COVID pandemic. And I don't want to put people in a position right now of having to bring people together if they feel uncomfortable with that. So this doesn't preclude that option. If you have, there are some groups that I know of that have teams that are actively working together, and they kind of have their own bubble. They know the precautions they're taking. They have an understanding with each other. If you have a group like that, like, you are more than welcome to have a weed wrangle this week with volunteers. Again, I'll include that in kind of an overall email that goes out to everybody. But we are also going to do kind of this social media driven kind of stay at home effort, just because until we have a better idea and a better handle on pandemic control, again, like I said, I really don't want to put anybody in the position of encouraging folks to come together when that may be uncomfortable for different people. So that's kind of that, and more to come there. I have a few notes out of First Coast Invasive Working Group this month. I wanted to give a shout out to one of their volunteers, Bev Lawrence, who at the conference a few weeks ago was one of the 2020 NASMA award winners. So Bev is in Northeast Florida. She's located on Amelia Island, and she is a retired U.S. Army Corps staff. She did permitting for the U.S. Army Corps, who decided she wanted to contribute in some way to her community and took on invasive species. So she is connected with the city of Fernandina Beach, as well as the First Coast Invasive Working Group, and is spearheading removal of Russian thistle, asparagus fern, and Brazilian pepper on Amelia Island, all of which are kind of, well, with the exception of asparagus fern, the Brazilian pepper and the Russian thistle are somewhat emergent. And so she really has just taken the lead and is keeping those projects going. So shout out to First Coast Invasive Working Group for nominating Bev, and to Bev for receiving this award. On a little bit of a somber note, I wanted to recognize Phil Gilliford, a volunteer who passed away this past month. He was with the First Coast Invasive Working Group, and he began volunteering for them back in 2006. So I actually worked with Phil at that time, and he was truly an incredible energy to be around. So dedicated, did probably more invasive species volunteering than anyone I've ever met. And he ran circles around us in the field. So when we had partner work days where we had FWC people, ourselves out there working, Phil literally just was nonstop. And so this news came out last week from First Coast, and I just felt like he deserved a shout out kind of on the larger scale, because he really dedicated a lot of his time for the last decade working on this issue in Northeast Florida. And so I just wanted to share that. And you know, he's in, his family and Phil are in our thoughts right now, because he will be missed. And he was just really an incredible volunteer. So with that, again, thanks everybody for joining us. We are here the fourth Wednesday of every month, except November and December. A reminder that past calls, including today's, are available on our website, and we are on social media. So Caitlin from First Coast is on our call today, and just wanted to thank us for giving a note to Phil. I mean, really, if you knew Phil, just an incredible individual. And so that's it for today. Thanks again. Look for some more CISMA updates from us in November and December, but otherwise looking forward to being back with everybody in January. So I hope everyone has a great rest of their year, and we will talk again soon. Thank you.